This is a production of Cornell University.
Right, so first up, I would like to say there are some sounds in my talk, so you might all want to be on your volume buttons to adjust as appropriate when they occur. I'll give you some warning. So can everyone see my slides okay? I'm going to shoot.
Looks good. Looks good.
Okay, great. Right, so I'm going to be talking about sounds of the soil today, and this is a collaborative project with Kyle Wickens as PI, myself as postdoc, and then we have three collaborators at Cornell, Holger Klink in the Lab of Ornithology, Greg McClaskey in engineering, and Johannes Lehmann in soil and crop sciences. So I'm going to focus here on below ground pests in turf grass systems, such as these white grubs. You can see the movie there on the right. So these are root feeding herbivores, and they cause extensive damage in turf grass systems by feeding on these roots. And so currently there's chemical management for these, which is to use blanket-wide pesticide applications. And often these are applied with no prior knowledge or assessment of species presence, population size, or location of those within that area. And so whilst they are successful in targeting the pests, they also impact other beneficial turf grass and soil organisms, such as pollinators and decomposers. And in addition to this, due to the temporal characteristics of many of these pests, many of these applications are mistimed or sometimes applied when they're just not necessary at all. So because of this, we have this need for a monitoring technique to see where and when the pest is present. So we can use manual scouting for this to assess populations. And this involves hand sorting and taking soil samples, but it's a time-consuming technique, requires training, and is also fairly invasive. So what we need is a monitoring technique that's non-invasive, cost-effective, and species-specific for managing pests. So passive acoustics is a very promising approach for this, and it fits all of those criteria. So acoustics is widely utilized to detect cryptic insects within crops such as grain, fruit, and timber already. And so we know that invertebrates produce sounds and vibrations both incidentally and actively. So I've put two examples up here on the slide. And if I just draw your attention first to the one on the box on the left. And so this shows the waveform of the sound with time along the x-axis and amplitude on the y-axis. And below that, paired in time, is a spectrogram. And so that shows the frequency composition of that sound with time on the x-axis and frequency on the y. So I'm going to play that movie now, and you'll see the red bar scrolling across. And this is the rice weevil inside wheat kernels making sound. So you should be able to hear a crackling sound there. And so other insects produce sound actively. So on the right, this is a scarab beetle that stridulates by rubbing body parts together to make this sound. And so if we focus on soils in particular, previous investigations with white grubs have yielded promising results in both field and lab settings using acoustics. But acoustics within soil is a lot more complex than other substrates such as wood because it's less uniform. And so we have few recordings of below ground organisms in soil. And so currently those two factors are limiting the utility and the application of acoustics for soil pest monitoring. So how do we measure sound and vibration in soil? Well, firstly, what are we talking about when we say sound and vibration? So here, if you look at this diagram, we have an insect here in the center, which is actively producing a sound. And you can see that sound is radiating into the air above, and that is what we would hear with our pressure sensitive ears. And it's also radiating down into the soil as well as a sound wave. In addition to this, the vibration of the body of this insect is traveling down the legs and is going into the surface of the soil in a substrate-borne vibration wave, which we would term vibration. And there are a number of different ways we can detect these mechanical waves in soil and in air. We can bury a geophone to measure velocity of vibration, an accelerometer to measure acceleration. And we can also use quite crude sensors like guitar pickups buried in the soil to detect vibrations. If we're feeling really swanky, we can point a laser at any reflective surface, and that will give us the velocity of vibration upon that surface. And finally, what we're all more familiar with is the use of microphones, which we can use for detecting sound in air, but we can also bury them in the soil to detect sound in the soil itself. Crucially, however, sound in soils is highly attenuated, and what that means is it loses energy across distance. And as I said before, soils are not uniform. They vary in compaction, particle size and structure, moisture and temperature. So taking these measurements in soils is a challenge. So the objectives of this project were to ID soil organisms and distinguish pests from non-pests using acoustics. And to do this, we'll develop a technique which we could then apply on a field scale to quantify distribution and activity of insect pests. And so there are two phases, lab and field, and I'll talk a little bit about the lab work to date. So we wanted to distinguish these organisms acoustically. So to do this, we had set up these turf grass and soil mesocosms in the greenhouse, and you can see pictures of those on the right there. And into these soil mesocosms, we added animals of different ecological functions. So first up, we added two root herbivores, that the larvae of these two species, which feed on the roots. Both of these are invasive. The crane fly is actually a relatively recent invader into this area, I think within the last 20 years. But both of these cause extensive turf grass damage. We also have mesocosms with engineers, earthworms, fungivores, springtails, and macro-decomposers, woodlice, pillbugs. And we also had control boxes without animals. And as for the mesocosms themselves, they had a uniform soil mix, which was the same across all boxes. We measured the moisture levels, and we added our animals at field realistic densities, and they were acclimated to the boxes prior to our experiments. So the sounds and vibrations that we're dealing with are extremely low amplitude, and because of that, when we took our laboratory measurements, we needed to enhance our recording conditions, essentially. So we built the following setup, which is essentially a semi-soundproof chamber within which we could put our mesocosms, shown here with in the center of that chamber on the right. And this essentially reduced the external sounds from the laboratory surroundings. And then we put this chamber on top of an air table, an anti-vibration table, which minimizes ground-borne vibrations from the building itself. So we had our mesocosms, and we recorded each of those inside the chamber for 15 minutes at a time, using both an accelerometer for vibration and a microphone for sound. And after those acoustic recordings, we also recorded our volatile signals, which were recorded by the Willett Lab. So what kind of sounds can we hear from those recordings? Well, I'll focus here on cranefly larvae. So I will play the top movie, and you should be able to hear some cracks and pops and crackles. And so you will have had some white noise in that recording as well. So we can reduce the noise. And so this recording below is a noise reduced recording. So the frequencies are slightly different and improved for our ear. And this is taken from longer term larval recordings. So how can we quantify those sounds? We can certainly hear that there's something going on. And I'll talk about this now, but the analysis is very much a work in progress. So I'm really just going to outline the techniques that we're using. So here on the top left, I have a waveform. Again, this is the cranefly larva. And one of the first things we can do is to calculate a spectra of that waveform. And essentially, if you think of a sound as a ball of wool or yarn made up of different colors, a spectra is a mathematical transform that takes out all of those colors and separates them out into respective frequencies in this case. So we can calculate the spectra of a sound across all of the sound, or just a fraction of the sound. And we get something that looks like this on the bottom right. And you don't need to worry about the numbers here, but essentially we have frequency on our horizontal axis, and we have spectral amplitude on our y-axis. And once we have these spectra, we can calculate simple mathematical parameters such as mean, median, mode of that spectra, and compare those both between and within our animal treatments. And how is that data looking? Well, there are 3 examples here. I've put up earthworm, cranefly larva, and control spectra. And here R is labeled the 4 highest peaks for us. And just to talk you through those, in red highlighted now, these are all peaks that are common between all the files. So that signals below 500 Hertz. But interesting, only in these 2 animal treatments, you see these purple shaded regions, and then in our cranefly spectra, you have a broader peak, which is not present in the other animal treatments. So that's just to give you an idea that pictorially you can see these differences, and then we can calculate metrics to quantify those differences. What else can we do? Well, we can look at the waveform itself. So here it is again, the same waveform, and you can see there are lots of vertical lines here. These are impulses. Short and sharp peaks of sound throughout the file. So we can measure the amplitude of those. So I've marked, the tip of each peak there in red. We can also set an amplitude threshold, and we can count how many of those peaks are above that threshold, which we would choose ourselves. And there may be more peaks in some recordings than others, and that might give us an idea of activity level or potentially species differences. Finally, we can look at the absolute value of the waveform, the positive part, which I'm now just showing in blue, and that's called the amplitude envelope. And so we can assess that for skewness and kurtosis, so a measure of symmetry and peakness, and together look at that kind of global shape of the envelope and how that may vary within and between our animal treatments. Finally, we can use an approach called automated acoustic template tracking. So basically, this is going through all of our files and picking aspects of the signal that we think may be representative of that animal. So here, I've just circled this as an example. We could use this as an acoustic template. And what we do is we go through a file and we mark how many times we think manually that template is appearing. And then we can use the same method, but automated. With machine learning, and so going through those files and seeing if we can automatically detect that template. And then we compare the manual and the auto techniques with each other, and ultimately aiming to optimize the automatic technique to be as effective as our manual marking when I listen to the file myself. And so you go on to optimize that automatic method and apply it to your raw recordings, which saves a lot of time. And ultimately to say, is this particular template only present in that animal treatment? So to summarize then, for more targeted below ground pesticide applications, we need a cost-effective, non-invasive monitoring technique. And passive acoustics shows great promise for this, and we're currently analysing data to see if different organisms can be distinguished using sound, and ultimately thinking about scaling this up to the field where you could measure sound across an area and maybe plot it in a similar way like a heat map, which I've tried to show in this diagram. Okay, thanks very much. I'm a little bit over time, sorry.
This has been a production of Cornell University, on the web at cornell.edu.